Yes, can do.
Super. Okay, so my talk is a bit different from what you've been hearing in the past sessions. So I work with natural polymers of bacterial origin and then use them in various biomedical applications. And I have to mention these are highly sustainable polymers and also very green. They are the few that can degrade in soil and in marine environment. They are recyclable, they are compostable. So really, I feel polymers of the future. So I have 4 different polymers in my laboratory: polyhydroxyalkanoates, bacterial cellulose, gamma polyglutamic acid, and alginate. Today I will mainly talk about PHAs because I have about 15 minutes and a little touch on to the bacterial cellulose so that we can cover a little bit of my detailed work. So this is a typical structure of polyhydroxyalkanoates. Apologies for those who don't like chemistry, but just to show you that here you can change R1, R2, and X values. So what we are dealing with is a polyester, but a family of polyesters. And what is really interesting here is that rather than having one property, one type of polymer, we're dealing with short chain length polyhydroxyalkanoates, or PHAs in short, which are normally rigid polymers, whereas the medium chain length polymers, which have a monomer chain length between C6 to C14, are really, really elastomeric in nature, very high elongation at break. And this is just a table. I'm not going to read it all out to you, but you can see the variety of properties in these polymers. Young's modulus going from a value of 0.008 gigapascal to about 3.5, elongation at break going from 1 to about 1,000. So I'm sure that gives you an idea. So this is the pilot plant in Sheffield that we produce these polymers using bacterial fermentation. And when I started many, many years ago working on these polymers, I focused initially on a group of bacteria called the gram-positive bacteria. The idea behind this choice was the fact that they do not have lipopolysaccharides, which are immunogenic in nature, very strong immunogens, and you don't want them in a polymer you use for a medical application. So I started with these, and this is a typical polymer, poly-3-hydroxybutyrate. Candice is now working on them with me. And here you can see it's a high melting polymer. This is a 3D printed strut. Glass transition is about 1.8, so quite low. Young's modulus is about 1,563 megapascal. Elongation at break about 4 or 5, not very high, and tensile strength about 19 megapascal. So quite rigid in terms of getting close to a gigapascal in terms of its Young's modulus. I also produce the elastomeric ones, the medium chain length PHAs, and these, like I mentioned, are very, very different. The melting temperature is much lower, about 53. I won't read out all the properties, but will point out that the Young's modulus is only about 9 megapascal here, and the elongation at break, in contrast to the low value you saw earlier, is about 600 or 550%. So a really elastomeric polymer. What do we do with these polymers? Of course, this is an additive manufacturing session, and processing is the focus. So you can see it can be processed in most traditional methodologies, including AM, which I will come to in a minute. And these are the technologies that we have used. So making porous structures, microspheres, gyrospinning them, 3D printing them, making tubular structures, melt electrospinning them, melt extrusion followed by laser ablation for drug-eluting stent production, and 3D molded structures that we have created. So this is just to show you that we can do FDM printing of poly-3-hydroxybutyrate and its composites, and I'll show you more about this later. And here is an example of an FDM printed medium chain length PHA, and you can see how elastomeric the structure is. So I truly believe that PHAs are a new emerging medical materials. They haven't really been used in a variety of things yet, but it's coming. And TIFA Incorporation, which is a spin out of Harvard University have obtained FDA clearance for this polymer or this polymer type. Of course, it's a family, and each time we use it for a different application, we have to go ahead and apply for an FDA clearance. But for sutures, they have already been cleared, and that's wonderful for my work because when we go to the FDA, hopefully soon, they are familiar with this polymer type. So this is what I've done in the last almost 25 years, frighteningly long, but they're very interesting polymers, and therefore I've been able to do all this with them. So I've looked at a variety of medical applications: bone regeneration, cartilage regeneration, kidney, semi-artificial pancreas, skin and wound healing, cardiac tissue engineering, controlled delivery of drugs, biodegradable drug-eluting stents, biodegradable nerve conduits for nerve repair, and now 3D models for disease. And the first one I hit was cancer, which I'm sure you'll understand the choice. So firstly, about bone tissue engineering. So this is the first example of 3D printing PHAs for bone tissue engineering. And initially, we wanted to look at the membrane around the bone, which is the periosteal membrane. And this is the first time ever medium chain length PHAs have been 3D printed. We, of course, had to optimize the printing conditions. I don't have to tell this to this audience: the temperature, the pressure variation, and of course, also the writing speed at which we printed. So here you can see some examples of 3D printed medium chain length PHAs. And the one that you see on the right is currently being tested in vivo in Spain as a bone replacement unit. We looked at the degradation. So in the body, we are using these scaffolds so that the cells can infiltrate, grow into the real tissue, and then this polymer disappears. So we looked at degradation, and indeed in 6 months, we observed that the MW value substantially decreased. And this is only in what is called phosphate buffer saline. If we really had conditions in the human body, I think it would be faster. And these are the temperature or the thermographs of the DSC, and you can see there are slight variations in the TG and the TM as expected with degradation. And last but not the least, we always have to check for cytocompatibility. So on the left, you see what is called indirect cytotoxicity, and the right is direct. So the cells are seeded onto the material, and these are bone osteoblast cells, and they look, as you can see, very green and healthy. Green meaning healthy, red meaning dead. We also looked at mineralization. As we know, bone is a highly mineralized tissue, and this assay told us it's an alkaline phosphatase assay, which told us that it was beautifully mineralized. And this you've already seen, but this again, I have to say, is a first successful 3D printing of P3HB. People have a huge difficulty because really the gap between it degrading and melting is very small, but we've managed to have some tricks up our sleeves, and that's worked. And the composite here with barium titanate. I'll quickly whiz through, I've got about 5 more minutes, I can see, some other technologies that we've used. This is gyrospinning along with Professor Mohan Edirisinghe at UCL. It's really a little cylinder with holes, and how beautifully we get these fibrous tissues, tissue-like structures, which are porous in nature, aligned. And we don't like killing animals, so we start checking our construct in an egg, where if the chick develops normally, we know that it is non-toxic, and do blood vessels grow around it, because it's always a challenge to get these scaffolds vascularized. And the red here tells us that it is nicely vascularized. The blue tells us that collagen or bone regeneration is occurring. When we checked this using an egg, then we went into a rat and again found beautiful results. So I won't go through this in details, but I'm sure the red and the blue tell you there is vascularization and there is bone regeneration. I'll move on and go closer to the heart, which is the cardiac tissue engineering. This was with a BHF-funded project. And the idea is that after a heart attack, the heart develops scar tissue. Just like when we fall, we develop scar tissue. And we were trying to replace the scar tissue, which is non-contractile, with contractile cells or cardiomyocytes. So we used stem cells, which you see on the right. And what we were trying to get them were to be these anisotropic, long, healthy adult cardiomyocytes. So we did something which is quite new in the area, which is melt electrowriting. And these are the sorts of structures we developed. This is an SEM. As you can see, there are long, thin struts. And this is actually with the medium chain length PHA, which is elastomeric and needed for the heart. And this is what we got, beautifully long, thin cardiomyocytes, which told us that we can use these melt electrowriting patches to mature the stem cells and replace the scar tissue with cardiac cells. We looked at the functionality, calcium transients. We looked at whether endothelial cells, which form blood vessels, can grow. And here it's a co-culture of the endothelial cells and the cardiomyocytes. This is the patch on a rat heart, and the rat heart accepted that patch as its own. We don't see any capsule formation. We then patched onto a real, into an animal, a patch with cells, and again, found beautiful integration. Just briefly touching on, I won't be able to go into the details. We've done now what is called a multi-material cardiac patch. Here, we encapsulate the cells in alginate. We bioprint them, so we are printing these cells with alginate. And you can see here the cells beautifully tolerate all this torture of going through a nozzle, and we get lovely green cells. We've optimized this. And then we use the medium chain length to act as a kind of a support for this alginate. Otherwise, it falls apart when we put it onto a heart. So this is a typical heart cardiac tissue patch. And this is the multi-material patch, which is alginate and PHA. I'll now move gear quickly. Antibacterial is very important. As we know, with COVID, antiviral, antibacterial are very important. And we have looked into printing antibacterial structures with PHAs. And here we are, we've got some structures. We've added antibacterial stuff to it. This is selenium strontium doped hydroxyapatite. And here you can see some preliminary printing results, nice composites, and they had a very good cell growth on them and also good antibacterial activity against Staph aureus and E. coli. Like I said, we are generating bioartificial pancreas by printing, which are releasing insulin and hopefully help diabetic patients. We are creating kidney filtration barriers for patients where the kidney becomes non-functional, again with printing. And we are creating a 3D stents, which is the drug-eluting stents. This was a European project where we did the melt extrusion and then laser cutting. But now we are looking at 3D printing and using Effie to kind of inform our design. Last but not the least, one slide on bacterial cellulose, that is the other baby in the lab. And you can see very beautifully nanofibrillated structures. And we've started 3D printing this. And this is a blend of bacterial cellulose and alginate. So I'd like to conclude today, hopefully on time, that polyhydroxyalkanoates are indeed an emerging class of sustainable polymers. You'll be happy to hear that we can get filaments out of these as well. There is a company that are producing PHA-based filaments now. And these are biodegradable, biocompatible, highly sustainable, and green polymers. You've seen how we can process them using a variety of techniques. We've used them for tissue regeneration, and the other polymer we've used is bacterial cellulose, but equally they are useful for any other bulk applications that that other speakers have been showing. I'd like to finish by thanking all the key scientists and collaborators, and of course the money without which this doesn't happen. My group in London, where I was until 2019, and my group in Sheffield, where I am currently. And thank you all for your attention. Thank you.
Thank you very much. Perfectly on time, which I also thank you for. I think it's really nice for us to hear, because we talk a lot about materials, and it was nice to get the contrast between the kind of Andrew's talk this morning, starting from the very first kind of materials that we were seeing in additive manufacturing, right through to some of these kind of latest examples. Now I'm just going to try to check whether we've got any questions in the chat or on with hands up. As always, for those of you who've joined us more recently, if you have questions, you can pop them in the text chat, or you can raise your hand and we'll come to you with questions. Just while I give people a chance to do that, I have a question for you, if that's okay. So you've talked a lot about the potential medical applications. What things is it that additive manufacturing opens up? Like, what does it allow us to do that we couldn't do previously, if that makes sense?
Yes, I think it's the complexity of structures to make bespoke structures. So, for example, in medical applications like wound healing now, we are thinking of doing in situ printing of the wound healing patch onto the patient directly in a hospital, rather than having to wait for the patch and, you know, delivering it, etc. And of course, 3D printing is the best way forward there. You know, you can take your machine in and print the structure onto the patient. And this is beginning to happen. And similarly for bone tissue engineering, again, additive manufacturing allows us to take the bespoke structure that is needed for the patient, and that's what you can do. So it is the control, the bespoke nature that really additive manufacturing brings forward, which no other technology can really provide.
Yeah, we've got a question in the chat. Now, this might be outside of your area. It's fine to say if so, but someone says they're working on 3D printing electroactive scaffolds. Do you have any suggestions other than graphene for materials that can induce conductivity in PCL scaffolds?
Yes, actually, there are quite a few other things there. So, for example, there is a polymer polyaniline that you can add, which can bring in electrical conductivity. You can add carbon nanotubes. You can use carbon blacks, like we are working with Candice on, to induce electrical conductivity. There are other polymers, P-dot as well, which you can use. So there are a variety of things that you can use to introduce electrical conductivity. And in fact, we're working with graphene, we're working with graphene oxide. I've previously worked with carbon nanotubes, polyaniline as well. So yeah, many options there.
That's great. I love when an answer is yes, there are other materials and there's plenty of them. We have time for one other question from in the chat. Thank you for a great talk. How much progress have you made on the intercellular communication in some of the bioprinted constructs? Or is the focus more on printing singular cells in a matrix?
Oh no, cells are very social. They want others. They want to talk to others all the time. They die if they don't do that. So all our constructs introduce intracellular, interaction, and we have a lot of attention to that, to have that, so that the porosity is, interconnected, so they can talk to each other and grow and become functional. That's crucial. You can't have them isolated in little compartments. They die.
Wonderful. So we are just still on track with our agenda, which I